for genetic engineering of crops for insect resistance. Why these insects become major threats to agriculture? So in order to understand why insects have become today major problem to agriculture, we need to understand the evolution of insects as well as plants. Plants also evolved during the course of millions of years. Similarly, insects also co-evolved during this period. So plants and insects have coexisted, co-lived together more than 350 million years. No doubt plants evolved initially, later on insects. But they existed and interacted for so many years. So here, plants provide resources, insects provide services. That is their true mutualism. Once this mutualism is somehow disturbed by different ways, by climatic changes or human intervention or plant evaluation or insect, whatever all the factors which contribute to this, they change this mutualism. Once the mutualism can shift, you can expect the detriment to either of the partners. So in that case, they try to overcome one's defense system by others. I will come to that a little bit later. What are the defense systems we come across in the plants as well as in. And again, here, because of this evolution, you see the huge diversity because of so many these selective pressures, whatever I mentioned earlier. And in response to this, the plants and insects also respond physically and as well as chemically. So now what are those? I will briefly explain. Is it okay, audible? Am I speaking properly? Dr. Khan?
Sir.
Is it okay, audible? Everyone is
yes, sir, it is audible.
Yeah, if any problem, please let me know.
Oh, yeah, sure.
Yeah, this is the evolution period where the insects and plants evolve. Now you see here the first land plant, algae, you see aquatic. This started during Paleozoic era. So that means more than 400 million years ago. From this algae, aquatic, Moses land plant, preliminary land plants, then ferns, ginkgo, conifers, cycads, and finally highly evolved plants, that is angiosperms. So it took millions of years from algae to become the angiosperms. So during this course of time, these plants, whatever evolved are today the so-called plants of angiosperm, they have undergone tremendous changes in the physical structure, morphology, anatomically, as well as florally, whatever different organs they are modified. Similarly, insects. See, the first insect group is Archaeognathata. This is the first insect which was present on the plants. So during this about millions of years, there was no major problem about the insects. But however, during this recent periods, during the last 100 years or so many insect groups have evolved, and finally the highly evolved insect group is Bacterium. And now most of our crop plants, crop production, in other words, damaged by these 3 or 4 groups of insects: Coleoptera, Lepidoptera, Diptera, Cooptera. These are the 4 kinds of different insects which cause significant loss to crop production. So keeping this in mind, Whatever, as I told you, there are number of insect species, thousands of insect species are abundant on the earth. All of them are not harmful to the crops. There are beneficial insects as well as some harmful insects. So whatever the insects which are causing damage to our crops are divided into 5 to 6 groups. The first group is the Leptoptera. So 37% of the total insects, they belong to this order Leptoptera. Followed by 26% of the insects belonging to order Homoptera. And 11% the insects come under Coleoptera. 3% Diptera, Mites 7%, and others 12%. So today we are more worried about the insects belonging to order Leptoptera and order Homoptera because they cause maximum damage to several crop plants. In order to maintain our sustainable needs for the growing population, we need to control these two groups of insects primarily. Because these two groups of insects are sometimes as much as 60 to 70% of crops throughout the different under different agroclimatic conditions. So this control of these insects become a major objective in plant growth. So what are the various strategies available to manage these insect pests on crop plant? First and foremost choice or preferable choice would be resistant varieties. So that means resistant varieties means they have the inbuilt characteristics of producing certain proteins so that the insect cannot attack those plants. So whatever the varieties developed so far by plant breeders, based on the resistant genes available within the primary genetic pool. So that means, for example, you take rice. You have hundreds of varieties of rice. If any variety has some resistant gene against a specific insect, you can make a cross and transfer that resistant gene to a susceptible variety. So that your variety can resist against that particular insect. Today we have many of the resistant varieties, no doubt. But unfortunately, we are growing only certain varieties in more or larger areas year after year. So with that, what happened? The resistant varieties have also become susceptible during this course of time. Now, the second option available is the insecticides or the pesticides. These are nothing but synthetic chemicals. Today, we are exclusively dependent on insecticides for controlling the major groups of insect pests. But similarly, insecticides also have certain lifespan. That means they can give protection to certain time. They may not provide same level of resistance for years, no doubt, no way. Because again here, people are using indiscriminately, and that means excessive pesticide people are using. With that, the insects are becoming resistant to that particular insect. So if you identify any new insecticide, that may give protection maybe few years. That also depends on how judiciously you use that particular insecticide. And then, and with the insects, the problem with the insecticides, the problem is, or chemical pesticides or chemicals, whatever you call, the problem is they contaminate the environment. And indirectly they affect the human health. So the excessive use of insecticides, they come into the environment. So people will be exposed to this contamination by insecticides and our health will be. So again, that is not desirable. Now the third aspect today, which I am going to talk to you, is about the transgenic plants. What are the transgenic plants? How these transgenic plants offer certain advantages over other methods. I will describe it briefly. And other than this, from insecticides, like transgenic plants, from insects side also, you have transgenic approaches to make them sterile. So if you release sterile insects, male insects, generally you don't get population. So these techniques also people have been exploited, but not that much success has been reported. So this normally also insects can be sterilized using radiations, UV radiation. That is one of them. But there you cannot identify which is male, which is female. So there is some practical problem. But transgenic methods, you can target the only male sterile insect. Okay, I am not going to today. I am not going to discuss that because that is a different aspect. Above all, finally, one has to follow integrated pest management. This is the best approach. Either you use insect transgenics, no doubt we have to use insecticides, no doubt we have to use resistant. If you combine all these with taking care of integrated pest management strategies, we can protect our crop plants better. So no single technique can provide you absolute resistance. So those, I'm sure most of you are familiar with this terminology. So what are GM crops or transgenic crops? Genetically modified crops are the product of introduction of specific gene or group of genes in a crop plant using recombinant DNA technology. So the gene or genes introduced may belong to either a distant species or a closely related species or the same species sometimes. So the gene can be anywhere within the available species on the earth. It may be from bacteria, it may be from yeast or fungus, it may be human or it may be animal or it may be a distantly related plant. Okay. So if you introduce those genes through recombinant technology and deliver or develop plants, we call them as transgenic crops. So what is the difference between transgenics and conventional crop breeding or non-transgenic crops? So in this case, conventionally they breed two varieties. So with that, wherever the sexual incompatibility is not there, they can make crosses and get hybrids or varieties, so that the variety B will be either having a desirable gene like resistant to insect or resistant to environmental factors, whatever it is. So here, what we are trying, we are transferring a set of genetic information to a recipient plant or the donor plant. So here we do not have any control how much genetic component we are transferring. Conventional approach. Whereas in transgenic approach, we know, define how much genetic material or genetic component we have. Is it a simple gene or a group of one or two genes? Like that, we know targeted genes which to be introduced. So initially, the first generation transgenic crops developed, they belong to either virus, insect pathogen resistant transgenic crops. I will give you examples of insect resistant crop plants developed using this technology, which are under cultivation for the past 20 years or so, more than 20 years. herbicide tolerant crops, abiotic stress tolerant crops. Still, we do not have crops which are growing in field conditions. Of course, there will be limited field trials have been completed. They may be anytime, they may be available. Improved nutritional quality. Yes, there are a couple of crops, but still we do not have that much under cultivation. And specialty crops for industrial and human health. Still, they are not under the cultivation. So my topic today mainly deals with the insect resistant crops. So that's what I mentioned. First generation crops, mostly they have the insect resistant genes as well as herbicide resistant genes. The second generation transgenic crops, here is the quality characters. For example, rice, beta carotene synthesis, because rice do not have beta carotene. So they introduced that pathway because vitamin A. Rice doesn't have vitamin A in the seed. So people now express vitamin A gene, rice industry. Third gen, they changed the complete metabolic pathway. So these are used for the, especially for industrial purposes. So whatever it may be, you see time scale. Initially, these transgenic crops developed around 1996. From that period onwards, now today we have number of transgenic crops for different types. So plant, as I mentioned, plant and insects, no doubt they interacted. At the same time, they evolved against each other in order to overcome their defense responses. So if you see the insect, so insect can change its behavioral and morphological adaptation. So these behavioral tactics against plant defense compounds. Plant, because the plant can synthesize certain compounds. So in order to overcome that defense pathway or defense mechanism, the insect can change its tactics or something like. For example, like avoidance. So if the particular plant is resistant, it may not attack. So it might change to another plant. Like that, avoidance. Utilization of alternative hosts. That's not instead of one host, it may go to another host. Increased consumption rate, change in feeding habits, development of specialized mouth parts to damage the plant at different in other parts of the plant. Instead of leaf, it may go to stem, it may go to root, like that, different. So for that, they change the specialized, they develop the specialized mouth part. Coming to molecular adaptations, so since the plant develop or produce toxic compounds, the insect try to sequester those toxic compounds. That way it can overcome that toxicity of those compounds. Then they also produce improved detoxification mechanisms. That means the enzymes, they can overproduce so that the detoxification can be improved, so that the resistance of that particular compound will be improved. Similarly, their digestive enzymes, modification. So in order to digest that target protein, so it develops alternate enzymes. So with that, it can overcome that the effect of that particular protein or compound. Similarly, plants, even they have adaptive protective structures. We all know that some plants have thick cuticle, trichomes, all these external factors. So these characters can prevent the attack of the insect. Similarly, plants also have molecular approaches against the insects. So plants can synthesize specific secondary metabolites which are toxic to insect. They can synthesize some class of proteins like lectins, hydrogenases, enzyme inhibitors, defense enzymes, protease inhibitors, like that. So these metabolites are toxic to the insect. So by changing their strategies, insect and plant, they try to overcome each other defense system. During this fight between these, sometimes insects become more prevalent on the crop. Sometimes the crop can protect themselves against these insects. So why all of a sudden this pest problem on crop? What could be the reasons? As I mentioned earlier, monoculture are growing the same crop in larger area. So that is one of the reasons for this pest outbreak. So growing crops as a monoculture that provides a large area of the pest favorite. So if you grow the same variety in thousands, millions of hectares, naturally the insect can definitely grow well on those plants and cause the damage. So that's why you have to always try to change the crop year after year. Not only that, our farmers they prefer certain varieties over others due to various reasons. So if you grow the same variety in larger area year after year, again that variety becomes susceptible to insect. Then, since we have irrigation facilities and plenty of fertilizers available and people are using herbicide, so when you apply this irrigation and fertilizers, the crop can grow very healthy and nutrient status also will be changed. With that, again, insects can be attracted more towards that, change their physiology of the plant. So all these factors contributing to the insect. Similarly, if you introduce any crop where there are no natural enemies of the pest, in the beginning I told you, not all the insects cause the damage to the crop. There are certain useful insects. So one of the useful insect is we call it as a natural enemy. That insect present on the plant, but it may not cause damage to the crop. So that causes damage to the insect which is causing damage to the crop. So if there are plenty of natural enemies on the crop plant, so the target insect may not cause that particular loss. So these natural enemies can take care of the target. And not only that, by introducing pest species, one geographic area to the other areas. So sometimes when we travel, people unknowingly bring some varieties of whatever it may be. If you introduce that variety in no environment where there are no natural enemies or there are no specific the target insect, once you grow them, so there again the pest species again become major problem. And by using these herbicides, pesticides, if you remove the natural enemies, because the insecticide works broadly on different insects. So when you spray insecticide, it not only kills your target pests which causing damage, but also other natural enemies which cannot cause any damage to the crop. Once the natural enemies are eliminated, again the insect can become major target. So these are the certain reasons for the outbreak of these insects. So when you want to use genetic engineering for the insect resistant crop plant, so one has to keep these 3 or 4 points in their mind. The first foremost important thing is, so for genetic engineering, we need a specific gene. That gene encodes a protein, specific protein. So that protein should have toxicity against given insect. So that means the protein which you are, or the gene encoding that protein which you are going to use genetically, give ample protection to the target insect. So now the first question is, can proteins compared to synthetic chemicals give you a level of protection which is economically viable? Because now today, as in my beginning I mentioned, we are using chemical pesticides, chemicals, agrochemicals. Some chemicals, no doubt, they are giving protection. So if you go for genetic engineering, the gene which you engineered, that was a protein, that protein should give the same level of protection or not. If the protein can give that significant level of protection, then only we can go for the genetic engineering. That is first. Then, whether the single gene is sufficient to give that level of protection, or more than one protein is required to get that required resistance. If that is the case, if you want more than, or if it is required more than 2 or 3 genes, then the question is whether those 2 or 3 proteins should have same type of mechanism of action, similar to the your chemical pesticides, or should have different types of mechanism? That is another important question. So preferably we need to have different proteins and different modes of action. They should not have same mode of action. Then they may not be that useful for the durable resistance or for the long-term resistance of plants. Then another major question: what level of expression is sufficient to cause that significant mortality of insects? So that means the amount of protein is crucial. Whether the nanogram of protein is sufficient to cause mortality, or whether microgram of protein is required, or milligram of protein is required. For nanogram of protein, yes, expression may not be a problem. Most of the genes definitely express that level. If it is microgram, slightly problematic. No doubt we can express up to microgram of protein. But once you need milligram of protein, then definitely that expression may cause some effect on the plant metabolism. So that should not, that level of expression should not affect the yield of your crop. See, why we are going for genetic engineering of crop sickness, insect resistance? In order to preserve the loss caused by the insect. At the same time, maintaining the original yield potential of the crop. Suppose by engineering your crop, if you produce only 50% of this original crop, there is no use. Although the crop is resistant, and compared to yield, it is not up to your control plan. Then nobody will prefer that. So the expression level should not interfere with the host metabolism as well as its host characteristics. Phenotypically, visually, there should not be any difference except resistance to the insect. Then only people prefer that crop. Another important, in how many years these insects can break that resistance. So again, how many years required to build up again resistant insect population on that particular crop? So now I will give you one example how BT crops performing on this particular aspect. So if that is the case, keeping all the factors which I mentioned, now come to the what kind of genes one can use for genetic engineering. I told you genes can be from anywhere, from bacteria to human being. So initially, the endotoxic crystal protein genes of Bacillus thuringiensis have been exploited for developing transgenic crop plants for insect resistance. So these genes are called trigenes, and there are 9 to 10 different classes of trigenes available. So all these genes are toxic to different groups of insects. Not all groups, but most of the groups are insecticides. Then similarly, vegetative insecticidal proteins like VIP1, VIP2, VIP3A. So you have triproteins as well as VIP proteins from prokaryotes. So what are these trigenes? So these trigenes have three domains: D1, D2, D3. These three domains are very crucial for the protein to cause insecticidal effect. If any of this domain is deleted, it will no longer cause the insecticidal activity. So one has to remember these three domains are essential, and in each trigene, these domains slightly vary. But however, these three domains should be intact, then only it causes the insecticidal activity. So what is this Bt toxin cause? How it causes the toxicity of the insect? So this is the Bacillus thuringiensis life cycle. So this sporangium, where you can see the crystal type of proteins are produced during sporulation state. They are in crystal shape, that's why they call crystal proteins. This crystal is dissolved in midgut of the insect. The insect midgut has high pH value. So that means these crystal proteins will be dissolved under high alkaline conditions. So once it dissolves, it becomes protoxin, that is activation in the insect gut. So that becomes 135 to 140 kilodalton protoxin, that is we call it as protoxin. But this entire toxin is not required for genetic engineering. We need only the N-terminal region of this protein, about 65 to 70 kilodalton. That is sufficient to cause the insecticidal activity. So for genetic engineering, people use only this around 65 to 70 kilodalton encoding protein. So during 1996, these three crops were engineered and released for commercial cultivation. Cotton, resistant to bollworm, maize, and potato. So whatever the Bt crops today under the cultivation, mainly the area occupied by the cotton followed by maize. These are the two predominant crops are growing in the field in various countries. So coming to plant side, do we have any genes which can be used for genetic engineering against insects? So from plant side, you have number of candidate genes. We have proteinase inhibitors, alpha amylase inhibitors. These are the abundant proteins present in different plants. Lectins, alkaloids, chitinases, tobacco anionic peroxidases, ribosome inactivating proteins, genes encoding secondary metabolites. Any of the genes can be used for genetic. Today, I am going to discuss with you about the one class of genes which we have been using in our laboratory. Those genes, they belong to class of lectin. And I will explain you why we are using these lectins for genetic engineering. So my next lecture will be on taking rice and cotton as a model crops. So what work we have done, I try to explain. So next few minutes I'm concentrating on sucking pest resistance on rice. Now, why we have to go for this sucking pest in rice? What are the major problems with these sucking pests? Now you see the beginning I mentioned during climatic change, because of climatic changes, human intervention, and other factors, they contributed by significant diversity of insects on each crop. So, for example, if you take rice as an example, this is the Green Revolution period, 1965. You all know. So, at that time, there were only 3 major pests on rice. That is 1965, 65 to 70 at that particular time. Now, today, we have more than 20 insect species which cause loss to the rice crop. So some of them, as I mentioned here, like BPH, GLH, WBPH. Similarly, small rice leafhopper, zigzag leafhopper, green rice leafhopper. These insects, they belong to Homoptera order. They come under sucking pests. Whereas stem borers, like this stem borer, then leaf folder, these insects, they belong to order Lepidoptera. In the beginning also I told you, comparatively Homoptera, the insects, majority of group insects, they belong to order Lepidoptera. For example, stem borer, which belongs to order Lepidoptera. The losses caused by stem borer on rice every year, that amount is equivalent to the food grain rice required for Bangladesh population. You imagine how much damage is causing this stem borer to the rice product. Similarly, the sucking pests like BPH, GLH, WBPH, which I mentioned, they also cause equal amount of loss. So if one can control these stem borer, leaf folder, and sucking pests like BPH, GLH, WBPH, you can minimize the rice yield loss to about 5 to 10% maximum to the other insects. So here, as I told you, again I'm saying here, the stem borer causes 29% of the loss to the rice grain crops. Stem borer alone. And these 3 insects, they belong to sucking pests, BPH, GLH. These 3 cause more than this 29%. These 3 together. So that means once you control these 2 classes of insects, you can save your rice crop without any problem. Now, unfortunately, those insects which belong to order Lepidoptera, like stem borer, whatever the technology we use or the genes we deploy, those genes may not be useful to control these insects. Similarly, whatever genes we have been using to control these sucking pests may not be useful to control the stem borer. Okay, all the transgenic technology, whatever the crops in cultivation, they were developed with BT gene technology. That means the tri-gene step. So those genes are useful only the insects which cause the damage by chewing, causing the damage to the plant parts. Whereas these insects, they suck the phloem sap of the plants or xylem sap of the plants. For control of these insects, the BT gene technology is not at all useful. So if you want to develop transgenic crops against stem borer, you have to depend on BT technology. If you want to develop transgenic rice against sucking pest, we have to depend on another class of genes. So here I will explain you how the other class of genes, that is a class of lectin group of proteins, are protecting or giving protection to these people. So if you see these insect damaged by this sucking pest, we call it as hopper bug symptoms. Now you see the crop, they become completely damaged. You see the hopper bug symptoms. This is this is the BPH, brown plant hopper. This is the WBPH, white bar plant hopper. You see the hopper. Once they attack like this, you don't get even single grain from that particular area. Not only that, these insects, they cause physical damage, number one. Second, they act as vectors for various viruses. That means insects cause damages in two ways. One is the direct damage like this, upper one. Other one is by transmitting certain viral diseases. For example, this rice tumbero virus. If the rice tumbero virus is infected on a rice crop, the total crop will be. So that means if you control insects, automatically you can also control the virus diseases. So that's why the insect genetic engineering of insects become major attractive trait in plant. Now, for the sucking pest resistance, what are the various candidate genes available? And what approaches one can deploy to make plant resistant to this insect? One strategy is overexpression of a candidate gene. So what are those candidate genes? Snowdrop lectin. This is called Galanthus nivalis agglutinin. Other one is garlic lectin. Allium sativum agglutinin. Soybean trypsin inhibitor. These are the candidate genes available for overexpression. So today I am going to describe you the two lectins, these manospecific two lectins have been deployed for genetic engineering of rice and cotton against sucking pests. The other alternate approach is RNA approach, RNA interference. So here what we do, we use the insect genes. We use the insect genes and then we silence that important gene of the insect growth and development so that the insect cannot develop. That way we can arrest the growth of those insects. So here we are using insect-borne genes in this particular approach. Anyhow, today I am not going to touch about this, but today I am going to cover you this snowdrop lectin and garlic lectin, how these lectins can provide protection to our crop plants against the. So those who have not seen, this is the Galanthus nivalis, that whatever I mentioned, GNA, Galanthus nivalis. See, ornamental plant. So normal using conventional breeding approach, you cannot transfer this gene from this plant to rice. No way. But however, through genetic engineering, we can transfer gene from this plant to rice. Similarly, this is Allium sativum, garlic, most of you know. Similarly, you cannot cross this to the rice crop. So we have used these two plants source for the genes of the lectins, and then we introduce it into the crop. What are the characteristics of these proteins? As I told you, they are manospecific. They bind the mannose residues of the insect receptors present on the midgut epithelial cells. And the GNA is the tetrameric protein, as in 4 identical subunits. And ASAL, garlic lectin, has a dimeric, having 2 identical subunits. Each is 12.5 kilodalton. They are highly insecticidal activities against important succubus. They are also potent and selective inhibitors of retroviruses, HIV, and cytomegaloviruses. Not only that, they are non-toxic. If you are using any protein for genetic engineering, it should not cause any effect on fungus. That is also important. So fortunately, these proteins do not have any effect on fungus. So one can use them safely to deploy into genetic. Now, coming to the genes we have used and the vectors we have used for generating the transgenic rice against such pests. So here I mentioned the GNA, non-reflecting gene, which has 480 base pair coding region. To drive this gene, we use the rice sucrose synthase 1 promoter. I am sure most of you are aware, for the expression of any gene, we need promoter. This is the promoter, 5' region of the gene. And we need to have terminator downstream of this 3' end of the gene. So this is coding region, this is the promoter driving the gene. And selectable marker genes, yes, we have used the herbicide resistant gene. This gene is driven by cauliflower virus, of course, NAS terminate. So why we use this rice sucrose synthase promoter? Because, as I told you, these insects suck the phloem sap. So this promoter, rice sucrose synthase promoter, it directs this GNA protein to the only phloem cells, phloem epithelial cells, so that the protein is readily available where the insect attack the plant. And not only that, it minimizes the expression of other tissues, because we want this protein should be available in the phloem tissue, because the insects suck the phloem. It should not be present in the seed, it should not be present in root, or it should not be present in other parts. So that way it reduces the metabolic energy to synthesize this food. If it expresses throughout the plant unnecessarily, the metabolic load is on plant and naturally other characters may affect. So to minimize that, we use the rice sucrose to drive this gene. Whereas in the case of garlic lectin, we use the constitutive promoter. It expresses everywhere. Because we are consuming the garlic, so it is safe to the human being. So that's why we use the constitutive promoter. Not only that, this particular gene also provides some resistance to other group of insects. That's why here we use the constitutive promoter. And of course, bar as a selectable marker gene. Dr. Khan, is it okay? Should I go fast? Is it okay for all the participants?
Sir, okay, fine, sir. It is fantastic, sir.
So can I go with the same level of this thing?
Yeah, sir. Sure.
If it is, I don't have time, you please let me know so that I can take care of that.
Time is there, sir. 25 minutes more are there.
Okay. This is the complete schematic representation of developing transgenic plant. So this is generally we use the rice callus as a target tissue. That rice callus will be infected with agrobacteria, carrying our desirable vectors, which I showed you earlier. So that after infection, we transfer to selection medium. Because we have used the bar gene, here we incorporated the phosphate the MS medium. So now you see some of the calli are actively growing. That means these calli are able to grow on this herbicide containing medium. Because in those particular calli cells, the bar gene is there expressing, so it will be inactivating the herbicide. Whereas other tissue, you can see they turn black. That means these are not transformed because they did not receive this particular gene, their cells. So the actively growing calli indicates that is putatively transformed. So that calli will be selected, regenerated using normal tissue culture. Then after proper rooting, they will be transferred to farms and grown in the net house under controlled conditions. Because we cannot grow them outside. So we have to grow them in the growth chamber or glass house or polyhouse. After 40 or 50 days of age of old of these plants, we test them for the herbicide tolerance of the plants. So now we dip these plants up to this mark, where you can see this mark up to this area will be dipped into the 0.25% of the Basta solution, that herbicide. So this is the untransformed plant without dipping in the herbicide. So naturally you don't see any damage. This is the control plant, untransformed plant, dipped in the herbicide. Now you see after 3 or 4 days, you see the complete damage of this leaf. And all these are different leaves from different independent transformations. Now you can see showing variable levels of tolerance. This particular result indicates that these are the putatively transformed. Now we have to molecularly characterize how many copies are present in the genome of these plants, or whether the genes are expressing or not. So transgenic plant means the protein should be present. Then only we call it as transgenic plant. I mean, by definition, even though gene may not express, we call transgenic plant because it carries the gene. But the functional meaning is the gene should be expressed. So those are the real transgenic plants. Now we do southern blot analysis to confirm whether the particular transgenic plant carry single copy or multiple copies. We don't need multiple copies of genes because they may undergo gene silencing mechanism. So if you have more than one copy, there is always possibility that the transgene may undergo gene silencing. Once it goes gene silencing, it cannot express. So you don't find any protein and no use at all. So for that, we initially analyze molecularly and we select single copy containing plants. Then we do for the Western blot analysis to confirm whether the protein is properly synthesized or not. Now you see this is the positive GNA protein, 12 kilodalton protein. Now in all these 4 plants, you can see the same polypeptide reacting with the GNA antibodies. So that means all these plants, the protein is expressed. Not only that, as I said, we use the rhizoprosynthase promoter to drive this protein to the phloem parenchymatous tissue. Now we see this protein, you see the GNA protein is restricted to this phloem and parenchymatous tissue. It is not there in other tissues. So that indicates the rhizoprosynthase protein promoter to drive this GNA to the phloem tissue. So whereas this control plant, now you see there is no such real analysis of plants come into it. Molecularly you characterize, everything is perfect. Now whether these plants could provide resistance, that is important. Now we evaluated these plants against brown plant hopper, green leaf hopper, white back plant hopper at DRR under the supervision of entomologists. The above picture shows the before infection. This is a 4-week old plants. This is for BPH evaluation. This is for GLH evaluation. This is for WBPH evaluation. All these are homozygous plants. I told you we need homozygous plants. That means T2 generation plants to evaluate the efficacy of any introduced gene. Now you see, after 15 days of infection, the controls, this one control and this line control, you see completely damaged. Now you see all the transgenic plants survived and growing normally. And here you see the control plant completely dried, whereas the transgenic plant is growing on par with the resistant variety. Similarly, white back plant of you see the controls completely damaged, whereas the transgenic plants are growing normally without. That means this test gives you that yes, the GNA protein is able to provide enough resistance against the genes. So here the beauty of genetic engineering is, in the beginning I told you we have many resistant varieties, no doubt. But none of the rice varieties are resistant to all the three insects. You have BPH resistant varieties, you have GLH resistant varieties, you have white back plant hopper resistant varieties. But none of them provide resistance to all the three. But here in the genetic engineering, just one transgenic plant expressing that GNA is able to provide resistance to all the three insects. So that's why the genetic engineering has become more popular technique for the developing transgenic plants against insects. Not only that, we evaluated under field conditions. So where we have to evaluate the hotspot areas, we have to go to hotspot areas. We did at Maruteru, which is in West Godavari district, which is the hotspot area for brown plant hopper. So now you see, here we have the control plant, transgenic plants, replicate trial. This is replicate trial. Now you see the one control plant plot I showed you, see the complete hopper burn causing damage to this particular plot, control plant. Whereas now in the close-up of this is the transgenic plots. Now you see the transgenic. You can see fertile grains. But however, you can see one here is sterile panicle. This is due to stem borer effect. Because these genes cannot provide resistance to Lepidopteran glucopteris. So that means in order to get broad-based insect resistance, we may deploy more than one kind of gene. I will come to that a little bit later. Similarly, we also engineered the garlic lectin. We got the transgenic plants, we confirmed them by using all the molecular mechanisms, herbicide tolerance, similar to GNA. Again, we evaluated these homozygous plants against brown plant hopper, green leaf hopper, white back plant hopper, using the T4 generation, stable lines here. Now you can see. All these transgenic lines are able to provide significant level of resistance to these three insects, like DNA, like snowdrop lectin. That means these two lectins would provide enough level of resistance against these three insects. Now we individually evaluated these non-human parts against these three pests. Now we want to combine these and see whether any additive effect will be there or not. So for that, we have different approaches for pyramiding these genes. One is sexual process. You select GNA expressing plant, garlic lectin expressing plant, make a cross. Get F1, F2, and then select the desirable combination of genes containing progeny, and then evaluate. That is one approach. Or sequential transformation. First you transform with one construct, again you retransform that plant with another construct having a different thing. Then co-transformation with multiple plasmids. Generally with agrobacterium, we can go for a couple of plasmids, not more than that. If you are using particle bombardment method, you can use many number of plasmids and co-transform and select the desirable combinations. Then use of single plasmids carrying linked genes like prokaryotic upper and lower. Expression of multiple proteins from a polyprotein. Fusion proteins. This is another kind of approach. So now I will explain you what we did in rice. One is the sexual process by pyramiding two genes, and also we use the fusion proteins to extend the broad range of response to this particular fusion. This is simple conventional method like assault transgenic line, GNA, F1, F2, F3, and then we confirm these plants carrying both genes by molecular analysis, PCR analysis. You see the F1s having both genes, and again F2, those having two genes were selected and then evaluated against again these insects. So here we used the pyramidal lines and the parents along with the resistant varieties as well as susceptible varieties. Now again you see the red graph, the red line indicates pyramidal line. So now you see on the pyramidal line significant reduction of the mortality of the insect. In all the cases, you see. So that means the pyramidal line, that means it has two genes expressing together. So these plants are able to provide significant reduction than their parents having single gene. No doubt, the single gene also able to protect. But when you have two genes together, definitely those lines are doing better. And this is required for the durability point of view. If the single gene is there, insect may develop resistance in, let us say, 5 to 6 years, depending on the condition. Suppose if you have two genes, then time required to develop resistance may be more than 5 years. That's why we need this pyramiding of genes, more than one gene we require. That's why we pyramided these two lines and develop durable resistance for insect, the sucking pests of the rice. Of course, entomotoxic effects. Now you can see the bean insect. That means significant reduction in the production of the insects. The fecundity was reduced. See on the transgenic plants compared to control plants. During this complete 21-day period, see on control plants about 350 insects were present, whereas it comes to on transgenic significant reduction you can see. Among all, again you see the pyramidal lines are showing significant reduction. So 350 versus around 50 insects. So many folds reduction in the number of insects on a plant. So naturally, here whatever the damage they cause minimum, it will be minimum compared to so many insects. And another important point here is, though there are some few survived insects, now you see the among the survived insects on transgenic plants, you see more of immatures than the adults. On transgenic, on normal plants, you see the more of adults, control plants, naturally. And few of them immatures. When it comes to transgenics, you see more of immatures. That means to develop to adult stage, it took longer time. And some of them could not reach adult stage. That means the protein has entomotoxic effects. So that is the another advantage of these transgenics. You see, very few are in the adult stage. So even though they cause damage, it will be minimal. And feeding ability. This we determine is an indirect way. So based on the excretia secreted by the insects, we determine the feeding ability. So here, feeding is directly proportional to its excretion efficiency. If it feeds more, it excretes more. If it feeds less, naturally it excretes less. So the feeding ability was reduced more than 90% on all the transgenic plants within 4 to 5 days compared to control. Though some insects are there in the previous slide I showed you, but they are not able to feed. So that way significant reduction of feeding was observed among the insects which are present on the plant. And these lectins, generally, the mechanism of lectin toxicity is they bind the mid-epithelial cells of the insect gut, midgut. There the receptors are present, so the protein binds there and causes the toxicity. So if we fed these insects on the transgenic crop expressing the lectin, and this on the control. This is the insect fed on control plant. This is the insect fed on transgenic, and we remove the midgut cells and seen the laser scanning confocal microscopy using the Western blot analysis of larval midguts to see. So the GNA, sorry, this is the garlic lectin. It has two receptors in the insect midgut epithelial cell. That's 60 kilodalton protein, one, and around 40 to 40 dollars daltons protein receptor. Whereas GNA, it binds to only 50 kilodalton receptor. It has only one receptor for the GNA in the BPH midgut, whereas GNA has two receptors. The pyramidal line, which has both the genes, now you see all the three receptors are present. Suppose if the insect, let us say, imagine theoretically, if insect may develop within 5 years resistance to the GNA because it has only one receptor. Suppose coming to the garlic lectin, it has two receptors. Insect may require more time to develop resistance. If you take the pyramidal gene, having both the genes, now you can, there are three receptors. It has to mutate in three different places. That may take more time. So that is the advantage of pyramiding these genes, so that you expect ear resistance as well as longevity, duration of the resistance. Similarly, we also attempted cotton to see whether the same genes can provide resistance to sucking pests. Cotton, you know the importance, I need not read all these things, but cotton crop is majorly affected by the Lepidopteran pests, followed by sucking pests. So here we have the two complexes on cotton, what we call as bollworm complex, that is the Lepidopteran pest. Among this, bollworm is the major pest, which cause significant loss to our cotton crop. For this, we have the transgenic BT cotton. The BT cotton can take care of this. And another one, quick bollworm. Mostly the BT genes are also giving some level of protection. But unfortunately, recently, this pink bollworm has developed resistance. So the BT cotton sometimes not able to provide resistance to, because these crops have only single gene. Remember, all the BT hybrids which we have growing now, they have only single gene. Now, another major pest complex is the sucking pest complex. Here, aphids, dacids, whitefly, millipede. These insects, they suck the sap of the plant and cause the damage. Not only physical damage, and also transmit certain viral diseases, as I mentioned in case of rice. So to control this, we have used the garlic lectin. Of course, this is a transformation protocol. Again, here we use the bar gene, selectable marker gene. Now you see the transgenic events are showing tolerance to herbicide. This is the control leaf showing complete death. Yes, we did molecular analysis, PCR, southern blots, and northern blots to identify the events required for the further evaluation. Now you see the jacid bioassays, untransformed control, and these are the two controls. One is susceptible control, other one is resistant control. And these are the four transgenic lines. Now you see at least these two lines are showing significant level of resistance to jacid. Now the survival rate you can see significantly reduced. Only the 9 to 28% of the insects could survive compared to 62% on controlled cotton plant. Similarly, fecundity, it reduced to 36 to 57% of the insects on during this 32-day evaluation period. Again, here you see Most of them are in the control you see most of them are adults, whereas on transgenic you can see most of them are in immature state. Only 41% adults were present on these plants compared to 70% adults on control plants, indicating the garlic lectin has some insecticidal effect on the jasper. Another second pest, white fly, which is again major pest on the cotton because of the introduction of BT cotton. Now these insects have become major problem. So to control these insects, people are using heavily heavily on insecticides. So again here these 4 transgenic lines. Sorry. Now you can see the significant level of tolerance. Control, and again this is one susceptible control, this resistant control. So these plants are on par with resistant control. Again now you see entomotoxic effect. Survival rate is only 18 to 20% compared to 80%. So indicating the garlic lectin is more effective towards white fly than jasper. Again here you see only 36% adults could reach compared to 81% of control. Now another approach we have taken up is one is pyramiding of the genes. Second approach is fusion genes. By combining the domains, this is called domain swapping. Domains of trigenes and domain of lectin gene. So we synthesize fusion protein in order to extend the post broad range. As I told you in the beginning, this is the D1, D2, D3 of the 3 domains of lectin, trigenes. And this D3 is almost similar to lectin domain. Here now we see D1, D2 of triproteins, and we use the lectin domain, carbohydrate binding domain of lectin. So this D3 is replaced by lectin. So this we call it as a fusion protein. That means fusion protein contains D1, D2 of triprotein and carbohydrate domaining of garlic lectin. We did extensive in silico analysis using different bioinformatic tools, and this is homology modeling. We docked against D3, docking of garlic lectin. So we replaced D3 with lectin, and we also did with the insect receptor, APN receptor, homology this, both with native triprotein as well as the fusion protein. Now you see the interaction of lectin is better than the D3 subunit of the triprotein. So in order to validate this, again here this interaction is shown clearly how D2 is interacting with this, domain 2 with this, and here the lectin interacting with the APN receptor. Now you see, with native protein, only 4 amino acids are involved with interaction with the APN receptor of the insect. In case of fusion protein, now you see 6 amino acids are interacting. That means more hydrogen bonds, more amino acids are involved in this. So that means the stability of the protein, fusion protein, is more than the native triprotein. Now, of course, here you can see the stability of this protein. The fusion protein is more stable and more favorable to the thermoenergetic. So we did all these bioinformatic tools and concluded that this fusion protein is definitely better than the native triprotein. This is the molecular surface interactions. You see, this D3 is interacting only this much area with the APN receptor. But here you see, there is no any gap, and also not only that, the area of interaction is more with the lectin. So our previous, whatever the data we generated is completely corroborating with this particular structure. Then we cloned the genes. This is the entire Bt gene, 1.8 kilodalton. And this is the 1.4 kb coding region that includes 1089 base pairs of D1, D2 of glycine, 375 base pairs of garlic lectin. So which comes around 1.4. Of course, here you see 1089 and 375 base pairs of garlic lectin. We express in E. coli. This is the native triprotein and this is fusion protein. We purified them and then Western blot analysis we did. It confirmed that purified proteins. And the purified protein is 60 kilodalton and native triprotein is around 70 kilodalton. This is the fusion protein. So this red letters indicate triprotein region. This blue region is the garlic lectin. Initially,
Sir,
we did.
speaker has come, sir.
Another 5 minutes. Can I take?
Okay, sir.
Just 5 minutes. Give me. Okay. Yeah, we did the artificial dibiosis against the Helicobacter pyogenes. Here you see the 0.5 micrograms is sufficient to kill this insect, whereas 1.5 micrograms of triprotein is required, indicating the triprotein is more entomotoxic. The fusion protein is more entomotoxic than triprotein. Similarly, for another insect, 0.25 micrograms is sufficient to kill this larvae, whereas tri1AC is required 0.1 micrograms, still it is able to grow. So that means it is better than the native triprotein for the against two insects. Of course, we did the ligand blot analysis to see the receptor binding pattern. You can see the fusion protein, you have the more of the receptors. Of course, this is the, as I told you, this is 8-fold higher than this, and this is 20-fold higher than the triprotein against these two insects. Then we express this in rice in order to see whether the rice plant can give protection to both Lepidopteran pests as well as Homopteran pests. We characterize molecularly. Of course, Western blot, PCR, molecular analysis. Then we evaluated a stem borer, which belongs to Lepidopteran order. You see, the two transgenic events are showing the inhibition of this insect growth on these plants compared to control. That means the fusion protein is able to give some level of protection to the stem borer. And this is the leaf folder. Again, you can see the growth inhibition of the Lepidopteran pests. So that means stem borer and Lepidopteran, the leaf folder, they belong to order Lepidopteran. So that means the fusion protein is able to give protection to these two insects. Of course, these are the different lines. Now you see the insects only on control, 2 could survive, 1 or 2 maximum, and here you can see. And of course, this leaf mortality, only remaining insects killed, only 1 or 2 present, and here you see majority of them are killed. And now we evaluate against BPH because it has the carbohydrate binding domain of lectin. So the same transgenic plants are able to protect against the BPH, that is brown plant, because the garlic lectin has already provided resistance. Now we put that in along with the D1, D2 domains of the tribe. So the fusion protein now able to protect even the BPH insect. Of course, now you see the feeding behavior significantly reduced on the transgenic plants. Of course, again now the survival rate, see significant level of reduction at the end of the bioassays period on the fusion protein. Again, you see these transgenic lines are showing the insecticidal activity against the brown plant order. Again, here you see the immatures are more than the adults, like the previous case. So all these results indicate the fusion protein is highly toxic, not only for the one group of insect, but for the more than one group of insects. Same way, feeding behavior also significantly reduced on transgenic plants. Again, we confirm the ligand blot analysis using the insects fed on transgenic plants, and we see there are 3 receptors which are definitely giving better protection than the single. Yeah, finally, I should acknowledge the support I received from Department of Biotechnology. Swarna Bharati Biotech Private Limited, Hyderabad. Without their support, I would not have done this. Thank you. Do you have any questions?
Thank you,
To
sir.
answer.
Tomorrow we'll have the question session. Today we have the next speaker ready with us.
Oh, sorry. Yeah.
Please, sir, you stop sharing the screen. Bhagirath, sir, you can start your.
It was a mute.
We are a bit late.
Hello.
I regret for the being we are a little bit late.
No, it's okay. No problem. Yeah, it happens many times.
The topic was very interesting and it was a flow.
Can you hear me?
Yes, you are audible and you can start sharing the screen, sir, now.
Okay. Okay.
Thank you, Rao, sir, for such a wonderful explanation. It was a nice flow of work you have presented with us. Really, it was very good work you have carried out. Tomorrow again, we'll be interacting with you.
Share. Yes, sir. Can you see? Can you see the screen?
Yes, sir.
Yes.
Yes, sir. I will be.
Okay. Shall I go ahead or?
Yeah, you can start, sir.
Okay. 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 This top.
Bhagirath, sir.
Yes. Okay. Sorry for this. Now you can see.
Yeah, first slide you have to start.
Yeah. So the word mycorrhiza is familiar to everyone. That is, myco stands for fungi and rhiza stands for roots. So it's an association between certain fungi and plant roots. And it has a very interesting history by itself. How the word mycorrhiza was coined. It was coined by a German botanist, by A.B. Frank, and he was a forest pathologist, and he was interested in going and collecting fungi from forestry species and then bringing it to the lab and then studying it. So once when he went to the forest, he found that roots of pine seedlings were covered with certain mantle of fungi. So he thought it must be a pathogen. So he brought the root sample to the lab, isolated the fungus, and then he could culture it. And then he took the healthy seedlings, pine seedlings, and inoculated them in order to prove the Koch postulates. So that, and waiting for that, the healthy seedlings will become diseased. But to his surprise, the opposite happened. That means the inoculated seedlings, which he thought will become diseased, they started growing much better than the uninoculated plants. Then he thought this must be a symbiotic fungus and not a pathogen. So that is how he coined the word mycorrhiza. In addition, he also added one sentence: this association between the plant roots and this fungi is symbiotic. So that is how we say that it is a symbiotic association between plant roots and fungi. Now we know that there are 4 different types, main types of mycorrhizal fungi: ectomycorrhiza, vesicular arbuscular mycorrhiza, ericoid mycorrhiza, and archid mycorrhiza. Whereas the ecto and ericoid are commonly found in temperate plant species, and the orchids, as the name itself indicates, is very strictly, obligately restricted to orchids. Whereas the one which is the second type, that is a vesicular arbuscular mycorrhiza, is the one which is commonly found in most of the crop plants, agriculturally important crop plants, and it is very common in tropical countries. And so this particular slide shows a transverse section of a non-mycorrhizal root. You can see this is a non-mycorrhizal root. You can see the epidermis, and you can also see. Pardon?
Slides are not moving. We are on second slide only. Mycorrhiza 4 types.
Oh, that's all. Now it is. You can see I moved.
Slides are not moving. Yeah, now they have moved. Third slide
Now
we are on.
it's okay. Is it okay? You can see that this slide with the transverse section of roots.
Sections we are seeing. Yeah.
Yeah. Okay. So the one at the top is the non-mycorrhizal root. And non-mycorrhizal root, you can see the epidermis, the outermost layer, the endodermis here, and then the cortex region. And the one in the left, what is below the left, is you can see the fungus is in soil. In the soil, it produces very large spores called as extramatricular chlamydospores. They are quite big. In some genera, it can go up to 450 micrometers. That means sometimes you'll be able to point out without the microscope. So big as that. And the hyphae enters the epidermis, penetrates the epidermis, and ramifies in the cortex region. It is restricted only to the cortical region. And in the cortex, it produces globose to oval structures called as vesicles, and highly branched hyphal structures called as arbuscles. And that is why they were called as vesicular arbuscular mycorrhizal fungi earlier. Now, some of the genera do not produce vesicles, and whereas all of them produce arbuscles. So that is how the word vesicular has been deleted. If you see the recent literature, they are referred to as arbuscular mycorrhizal fungi. Earlier literature, if you see before 1990s, then you will see it's called as vesicular arbuscular mycorrhizal fungi. And next slide. So if we just go out and in the college campus or the university campus and then pull out any plant in Nizamabad, in your college campus, try to bring the plant roots to the laboratory and try to strain it with trypan blue. As you can see in this particular slide, you can see that the root is filled with these arbuscular mycorrhizal fungus. What you are seeing, the globular structures are all vesicles. So very, very characteristic feature. And the next here, we can see the arbuscules. Arbuscules are highly branched, dichotomously branched hyphal structures, and they are short-lived. Just 4 to 5 days is the lifespan. They undergo degeneration, and then we don't see them clearly. So, because they produce vesicles and arbuscules, that's why I told earlier they were called as vesicular arbuscular. Now, couple of genera, 2 of them do not produce vesicles, and now we are calling them as arbuscular mycorrhizal fungi. And all these fungi belong to the phylum Glomeromycota. And Glomera mycota has 3 classes and 5 orders, 14 families, and 26 genera. Okay, 3 classes, 5 orders, and 26 genera. And then they have 230 described species. And of course, this is the classification by OL 2011. And we do have certain other classification also. Uh, so it slightly varies from the taxonomist to taxonomist. And this is the one which is very, very commonly followed. And I said there are 26 genera of fungi. And if you ask the next question, of these 26 genera, which are the most common ones? And the most common genera are these, the 4 genera. A is Glomus, B is Gygospora, C is Aculospora, and D is Entrophospora. So these are the 4 most commonly occurring fungi throughout the world, including Indian soils. So these are the 4 genera more frequent. And of these 4, again, if you ask which is the most frequent, A is, that is Glomus, is the most commonly occurring genus. And these fungi are all obligate symbionts. That means we cannot grow them in a laboratory medium like potato dextrose agar, Zipex medium, or Richards medium. They need the roots of a living host to grow and multiply. So this is how we maintain the different species of fungi in my institute. I can say each row is one species of fungi which is maintained in the glass house. And this is a slide which is taken, I think, in 1970s. Okay, so here also I want to say some historical background about it. So it was in 1970s, I got a Fulbright fellowship to go and work in University of California. And when I went there, of course, the work which was assigned to me was to transfer the NIF gene, the nitrogen fixing gene, from Rhizobium to a saprophytic bacterium, Herba spirillum. So this was the type of work which I was supposed to do there. So when I went there, I started the work, but the next lab was working on mycorrhizal fungi. So in the first week itself, I used to peep into the next lab. Somehow, what the next lab working on mycorrhiza, what they were doing, somehow interested me more than what I was supposed to do. So I went and asked the professor, Professor Mortimer Starr, who was the editor-in-chief of the annual reviews of microbiology, and I went and asked him, can I shift to the next lab? Then he said, you have come on a Fulbright fellowship. Fulbright office is in Washington, D.C. You talk to those people, and if the other professor, the mycorrhiza professor, has no objection, I have no objection. So I called the office in Washington, D.C., and the lady in the office took the phone, and she said, if both the professors have no objection, we have no objection. That is the reason I shifted to the next lab in a very short period. So I started working on mycorrhizal fungi. This Fulbright fellowship was for 1 year. And it was December 31st when I came, then the fellowship was to end. So sometime in October, November, the professor asked me, if you want to continue in my lab, so there is no problem, we can take you on contract basis for 5 more years and you can continue your work on mycorrhizal fungi. So I talked to some of my Indian friends who were in the university campus there, and they all suggested that I should come back to India. The reason they gave was very interesting. They said we couldn't get a good job in India and we are here struggling as postdocs or on contract basis, whereas you are already an associate professor in a university, government university, and you should go back. So that is how I decided to come back. When I came back to India, scanning through literature, I could see that very little work or practically no work has been done on mycorrhizal fungi, excepting for some work mainly on ectomycorrhizal fungi, that was done in the ICFRE in Dehradun. And so when I came back, I just told the students, my postgraduate students, go and collect the root, our crop plants, bring the root samples, let us see whether they are mycorrhizal. So they brought varieties of plants and we stained the root samples and we found most of them were mycorrhizal. So this is one of those early slides taken in 1970s. So the interesting part here is many of them became the world first report because the Western world, they do not grow the crops what we grow here. They do not grow finger millet, they do not grow cardamom, they do not grow clove and so on. So many of them became the world first report. And then we also took the soil sample and then saw whether we have the spores. And we could see that plenty of indigenous mycorrhizal spores in our soil. When I was coming back from University of California, I talked to my professor, Dr. John Mengi, and asked him, he asked me, what gift shall I give you? Then I told him, the best mycorrhizal fungus you have. And they were giving the best mycorrhizal fungus to the farmers who were growing oranges, avocados, apples, and so on. So that best fungus he gave me. So I brought that fungus and we wanted to see how our plants respond to inoculation with this best exotic fungus. And so this is one of the first experiments we did. This is with Bengal gram. And this we did with autoclaved soil. You can see the difference here. Because it was autoclaved soil, no indigenous mycorrhizal spores were there or fungi inoculum was there in the soil. So this much of difference was there due to inoculation. The next thing we wanted to do, use the natural soil which has the native endophytic fungi and see what sort of difference we are going to get. So this is one of the very early experiment with the natural soil. You can see the difference in groundnut. So this particular person, which was actually a PhD student's thesis work, and he did his work on groundnut. And soon as he finished his PhD, he got the microbiologist position at ICRISAT in Hyderabad. And so he was very happy, and then he went there. I have included a few more slides. This is the difference we get in case of cardamom, and this is the type of difference we get in case of neem. I just included 3 slides. One, an agriculturally important crop. One, horticulturally important plant. Other one is a forest tree species. So when we inoculate with an efficient fungus, this is the type of response we get due to inoculation. The next question. And what is the mechanism? How it improves plant growth? And here, if you just take the first example of cotton, you have uninoculated versus inoculated. We can say there is significant increase in dry weight. And also look at the phosphorus, zinc, manganese uptake. Again, very significant increases in the uptake of these nutrients. So it's very clear that the mycorrhizal fungi help in the uptake of nutrients. The next question, what type of nutrients it helps? Again, we have to understand that there are 2 different categories of nutrients present in soil. One is the nutrients which move very freely in soil solution, which we call as nutrients moving through mass flow. Nitrogen, potassium are examples of nutrients which move very freely in soil solution. We call them as mobile nutrients. There are some nutrients which do not move freely in soil solution. They are called as immobile nutrients or diffusion limited nutrients. Examples are phosphorus, which is a major nutrient, zinc, copper, etc., which do not move freely in soil solution. So with this background, let us see this particular slide. This is a root sample, and all of us know the plants take the nutrients through root hairs. So here in the root hair region, there is some available phosphorus. Whatever phosphorus is present in the root region, the plants take. And as I told earlier, plants take phosphorus in the form of H2PO4 ions. So all the H2PO4 ions that is present in the root hair region has been taken by the root hairs. But phosphorus is an immobile nutrient. There is plenty of phosphorus away from the root hair region, but it is not able to move to the root hair. And it's here the mycorrhizal fungi play the most important role. They travel a much longer distance compared to root hairs, scavenge a larger volume of soil, take the phosphorus and pump it directly into the host, thereby short circuiting the nutrients. Okay, so this is how the mycorrhizal fungi help in the uptake of immobile nutrients like phosphorus, copper, zinc, and so on. And what are the other mechanisms by which they help plant growth? Being a microbiology lab, we were interested in studying the interaction between mycorrhizal fungi and the nitrogen-fixing bacterium Rhizobium japonicum in soybean. And this was a field experiment. Again, I would like to say most of the mycorrhiza workers were very happy with pot culture studies done in a glass house. Very few studies were done under field conditions. So this is one of the very early field experiments that was conducted with the mycorrhizal fungi and Rhizobium. What is the result that came out of this experiment was the synergistic interaction between mycorrhizal fungi and the nitrogen fixing bacterium Rhizobium. What I mean here is dual inoculation resulted in much higher dry weight and the grain yield of soybean. What is it due to? I told that there is a synergistic interaction between these two organisms. The mycorrhizal fungus increased the number of nodules and nitrogen fixation by Rhizobium. And Rhizobium in turn increased the percent colonization and phosphorus uptake by the mycorrhizal fungi. Thereby, the dual inoculation resulted in better growth and nutrition, and thereby better plant biomass and grain yield. So this was followed by an interaction between a free-living nitrogen-fixing bacterium Azotobacter and the mycorrhizal fungus. The result was the same. That means a synergistic interaction between the two organisms. And then we started studying the interaction between mycorrhizal fungus and a phosphate solubilizing bacterium. And here I have to mention that there was a controversy amongst the mycorrhiza workers. Some mycorrhiza workers felt that the mycorrhizal fungi can solubilize unavailable form of phosphorus and release the H2PO4 ions, which the plant roots can take. But other 50% of the mycorrhiza workers did not believe this. They felt that the mycorrhizal fungi never solubilize the unavailable form of P. So in this particular experiment, we use P32 labeled tricalcium phosphate and did this experiment, and which the results very clearly showed that mycorrhizal fungi do not solubilize unavailable form of P. So in that case, what can we do? There are organisms which can solubilize unavailable form of P, which are phosphate solubilizing bacteria and fungi. That is how this interaction study was done. So again, we could see a synergistic interaction between the mycorrhizal fungus and the phosphate solubilizing bacterium, which I will explain in this next slide here. Say this rock phosphate, some of the farmers use the rock phosphate as a source of phosphorus, which is not an easily available form of phosphorus. And in the soil, if we add phosphate solubilizing bacteria, they act on the rock phosphate, produce organic acids, and release the H2PO4 ions, which is taken by the mycorrhizal fungus. So that is how, again, we have that the dual inoculation is much better than the single inoculation with either of these two organisms. Another aspect that mycorrhizal fungi help in the biocontrol of plant pathogens. And this was again the first PhD student's work in groundnut. In groundnut, we have a serious disease caused by the soil-borne pathogen Sclerotium rolfsi. You can see this groundnut plant and how diseased it looks. Diseased with the pathogen Sclerotium rolfsi. In the third one, plant, we have introduced the same amount of pathogen plus mycorrhizal fungus. You can see the difference between these two. And you can definitely say that mycorrhizal fungus has helped to alleviate the severity of the disease caused by the pathogen. So we went one step further and see what is it that present in the mycorrhizal fungus that prevented the soil-borne, eliminated the soil-borne pathogen. So we found that the roots of mycorrhiza, the mycorrhizal roots contained higher concentration of OD phenols, orthodihydroxyphenols, which inhibited the germination of the pathogen. So which clearly answers how the mycorrhizal fungus helps in alleviating the pathogen. And the next thing is, again, in and around Bangalore, we have a lot of severity of the nematode diseases. So this is again, especially in tomato or any other solanaceous crops. This is a solanaceous tomato root, and you can see that we introduced about larvae of Melodagain javanica. You can see the tertiary galls called by the nematode. Here, the right side, we introduce the same number of larvae of the nematode plus the mycorrhizal fungus. So here you can see a bit difficult to see. It produced very small primary galls and it did not develop into secondary or tertiary galls. So we were really breaking our head how to interpret the result. We had discussion with our nematologists. We had scanning through literature. Finally, we could find that sulfur-containing amino acids prevent the development of nematode larva into adult. So this gave us a clue because the nematode larva penetrated the tomato root and formed the primary gall, but it did not form the secondary and tertiary roots, which very, very clearly showed that it prevented the development of larva into adult. Thereby, we do not have these tertiary galls. So again, what could be the reason? And we found that in the roots of tomato inoculated with the mycorrhizal fungi, the sulfur-containing amino acids were higher. And scanning through literature, we could see that sulfur-containing amino acids prevent the development of nematode larva into adult. So this is the re-explanation which we could give. And now I am just summing up the various mechanisms of improved plant growth due to inoculation with AM fungi. One is increased nutrient uptake, especially the diffusion limited nutrients. The second is synergistic interaction with the beneficial soil organisms like nitrogen fixers, P solubilizers, PGPRs, and so on. Then it's also protecting the plants against plant pathogens. And better drought tolerance also helps the plants to overcome water stress. We just very recently completed a project funded by ICR where we have shown at the field level that it helps in overcoming water stress. And it also produces the growth promoting substances like cytokinin, indolactic acid. So it's a cumulative effect of all these which makes the plants to grow much better. And now this is another aspect which I want to emphasize. All the workers, the Western workers from UK as well as from USA have been saying that mycorrhizal fungi are not host specific. What do we mean by this? We take isolate the mycorrhizal fungus from wheat crop, inoculate banana, it will colonize banana root. Take it from banana, try to inoculate neem, it will colonize neem. So that is why we call it as not host specific. It is true even today, but this was one of my students' work where he found that the one which is the last one, the first one, is uninoculated control. And he inoculated the Lucina leucocephala plants with different mycorrhizal fungi. And from this, we felt that the last fungus is the best fungal partner for Lucina leucocephala. So we proposed a new hypothesis. Though mycorrhizal fungi are not host specific. They exhibit host preference. That means for a particular host, there is always the best fungal partner. That means we can, suppose your crop of interest is tomato, we can screen various mycorrhizal fungi and select the best mycorrhizal fungus for inoculating tomato. So this hypothesis, when we proposed, somehow Western world did not agree. So there was big opposition from US as well as UK. So we have to show it in many crops. This is in case of cashew. See, the last one is the best mycorrhizal fungus for inoculating cashew. And this is citrus rootstock. You can see the one in the middle here. That is the best mycorrhizal fungus. So when all these things were going on, the United States Department of Agriculture came and they said, if your hypothesis of host preference is true, we are giving you a project for 5 years. What we want is at the end of 5 years, best mycorrhizal fungus for inoculating Lucina leucocephala. Okay, so the money came in dollars. Our university was very happy. I was very happy because I could help many students and with the fellowship. And then we started the work. And what came out of the 5 years, I have put in one slide. So, and we have, so the best mycorrhizal fungus for inoculating Lucina leucocephala is Glomus mossiae. And so there are 4 different cultivars of Lucina leucocephala that is cultivated in Karnataka state. So we selected the fungus which will work on all the 4 cultivars, because if one forest nurseryman or a farmer asks for the culture for inoculating Lucina, I can't give one culture to him, another fellow growing another variety, I can't give different culture to him. So we wanted to give a same culture to all the Lucina growers. So that is how we selected the best fungus which works on all the cultivars of Lucina leucocephala. And when this work, and this is the difference in the root system, you can see, and these are the 4 varieties, cultivars of Lucina, K8, K28, K67, and 72. And working in an agricultural university, once in a year, we have to face the farmers. Krishi Mela day comes. We have to face the farmers. I tell you from my personal experience, it is much easier to face the students. It is much easier to face the scientists. Sometimes it's much more difficult to face farmers because they ask such intelligent questions. So when this Krishi Mela was going on, one farmer asked, which is the best mycorrhizal fungus for inoculating Lucina? I said, Glomus mossiae. He came up with the second question, which is the next best fungus, which I never expected. So I told him it is Gygospora margarita. Third question from him, what will happen if I mix both these fungi and inoculate? Then I said, sorry, I don't have any experiments done on this aspect. Please come next year. I will do this experiment and give you the result. So we did this experiment and there was no adverse effect on mixing those 2 organisms. They were not antagonistic to each other. So you can as jolly well mix both the organisms and give to the farmer. In fact, it may be better in one soil is more favorable for mossiae, other soil may be more favorable for Gygospora margarita. It will work very well. And when this work was going on, the Director General of ICFRE, that is the Indian Council of Forestry Research and Education from Dehradun, happened to visit the university. And it was somehow, whenever any visitor comes to the university, the Vice Chancellor of the university used to bring them to 2 departments, crop physiology and microbiology. So the DG of ICR also was brought to microbiology, and I took him to the glass house and showed him the difference due to mycorrhizal inoculation. And he was surprised and he said, are you sure that your tiny little fungus can bring this much difference in plant growth? Sir, what you are seeing, you have to believe. So then he said, yes, we are going to give you a big project. What we want is the best mycorrhizal fungus for inoculating various forest tree species. Another project came, we selected, I just given one photograph here. This is teak wood, Tectona grandis. You can see the difference in the growth of the teak plants, seedlings. And so likewise, we have inoculated, we have screened and selected the best mycorrhizal fungus for various forestry species. I have just given you the list, some of them here: Acacia nilotica, Acacia auriculaformis, Acacia holoceracea, Albizia lebec, Azadi recta indica, Caliandra, and Dalbergia sissu, and Tamarindus indica, and Tectona grandis, and Dalbergia latifolia, the rosewood, again a very costly wood. And at this time, the Department of Horticulture people, they saw the results in the forestry species, and they said, we want the best fungus for inoculating various horticultural plants. Another couple of projects came from horticulture department, so we could develop the best mycorrhizal fungus for inoculating various horticultural plants like tomato, capsicum, coffee, cardamom, including flowering plants like marigold, chrysanthemum, and ornamental plants, and aromatic plants like rosemary. And this time, the National Medicinal Plants Board came and they saw this, our publications, and they said, we are giving you a project, and what we want is the best mycorrhizal fungus for inoculating various medicinal and aromatic crop plants. So we have developed the best mycorrhizal fungus for inoculating various medicinal plants. Having selected the best mycorrhizal fungus for inoculating various plants, what next was the question. How it's going to be useful to the end user, that is the farmer. So for that, we developed this phosphate response curve. So in case of x-axis, we have the increasing level of phosphatic fertilizer. In the y-axis, we have the plant biomass. And we are taking the example of a mycorrhizal plant and a non-mycorrhizal plant in this experiment. So let us take the non-mycorrhizal plant. To produce that amount of biomass, how much of phosphatic fertilizer it requires? Let us say 70 kg. And the mycorrhizal plant, to produce the same amount of biomass, how much of phosphatic fertilizer requires? Let us say it is 50 kg. That means we are saving 20 kg of phosphatic fertilizer. This is what we could show through this phosphate response curve. We have taken to the field and we have done this with various crop plants, and I've just included one slide, next one. This is in case of chili. The recommended level of phosphatic fertilizer to chili is 75 kg P per hectare. And with the uninoculated plant, given the recommended level of fertilizer, P fertilizer, what is the yield? It is 1.81 kg per plot. And we had the other treatment where we had 50% of the recommended phosphatic fertilizer, and we have tried it with various mycorrhizal fungi. See the last one, that was the best mycorrhizal fungus, and with 50% of the recommended, look at the yield. So which very, very clearly showed. We have done this with capsicum, we have done it with tomato, and we have done it with many crop plants, and we could show very clearly 50% of phosphatic fertilizer can be saved through inoculation with selected fungus. Nowadays, everything is high tech. Okay, so agriculture is also becoming high tech. At least I know in and around Bangalore, farmers do not raise their own seedlings. They go to a nursery man and buy the seedlings. The nursery man raises the seedlings in pro trays or root trainers just like this, and the farmer goes and buys the seedling and plants it directly in the field. And here, the next slide is the close up. So this is how the seedlings are grown. And it's so easy to mix the mycorrhizal inoculum with the substrate, and the roots are colonized by the introduced mycorrhizal fungus, and the farmer plants it directly in the field. Very, very simple technology. And so this is becoming very popular around Bangalore. Now we are concentrating mainly on this nursery technology, and so it's becoming very, very popular. And at this time, the Department of Horticulture people again came to us and said we have a problem in citrus rootstocks. What is the problem in citrus rootstocks? They stay in the nursery for 24 months, for 2 years. Then the stem should reach the pencil thickness when it is ready for grafting. Okay, so it takes 24 months for it to reach the pencil thickness. So they came and said, please see whether the mycorrhizal fungus can reduce this period. So this was again given as a thesis problem to one of the students, and this is what he did. The results: A is uninoculated, inoculated with the different mycorrhizal fungi. See the last one. So it reached the pencil thickness in 18 months instead of 24 months. So thereby saving 6 months of the seedlings remaining in the nursery. So which is a big advantage for the citriculturists. It's a very common technology followed by the citriculturists now. And in and around Bangalore, floriculture is a major industry. In fact, a flight full of flowers go to Amsterdam at least twice or thrice a week. So those floriculturists came and then said, can mycorrhizal fungi help in the flowering plants? So we have done it with many flowering plants. I just included one slide. Here in case of chrysanthemum, inoculated versus uninoculated, you can see the difference in flowering. But one more thing I have to add here, the inoculated plants flowered 3 weeks early. Somehow the floriculturists were more interested in that than the number of flowers. So they were said, oh, very good, that is flowering 3 weeks early. And at this time, the post-harvest technology department, we have a post-harvest technology department at the university, the professor, she came to me and said, can I take these flowers, both from mycorrhizal as well as non-mycorrhizal plants, and then keep it in the flower vase and see whether how long they stay in the flower vase. I said, ma'am, you take and see. And she came and told, your mycorrhizal plant flowers stayed 3 days more in the flower vase. I was really surprised. And now we have the several reports from other countries that the mycorrhizal plants stay 3 to 4 days more in the flower vase. And at this time, again, the horticulture people came to us, said we have a problem in cashew. What is the problem in cashew? They said the nurserymen raise the farmer raises the seedlings in polybags and he plants it in the field. When he plants it in the field, there is nearly 40% mortality. So if he plants 100, 40 of them die, only 60 of them survive. So this is what is commonly called as transplant shock, which occurs in some other crop plants. So this again, another project came. They said whether you can overcome this through mycorrhizal inoculation. And this experiments, we did it we have a cashew research station near Mangalore. So all the things were done in cashew research station. And this is the result. You can see the plants, seedlings inoculated with these two fungi, Aclospora laevis and Glomus etunicatum. When they were planted in the field, there was 100% survival. The uninoculated plants, when they were planted in the field, there was only 63% survival. Again, a technology commonly followed by cashew growers now. At this time, the tissue culture people came to us and said, how will our micropropagated banana respond to mycorrhizal inoculation? So this is what we did. Inoculation with the mycorrhiza was done at the time of hardening of the plants, and you can see the difference in plant growth. And this is again a tissue cultured Ficus benzamina. Here I wanted you to see the first one, inoculated with only mycorrhizal fungus, and the last one, there we have inoculated with a microbial consortium. The same mycorrhizal fungus, we have included a mycorrhiza helper bacterium, Bacillus coagulans, and a biocontrol agent, Trichoderma harzianum. So from this, it is very, very clear a microbial consortium works much better than inoculating only with mycorrhiza. So now our trend is to develop microbial consortium rather than use only mycorrhizal fungi. Again, few slides. Compared, this is in case of Acacia auriculaformis, uninoculated, only inoculated with mycorrhiza, inoculated with the consortium. This is a project work which we completed, project funded by DBT, and you can see in case of tomato and other solanaceous crops, tomato and capsicum especially, we have a wilt disease complex. Why it is called wilt disease complex? It is mainly because that it is caused by more than one pathogen. We have a fungal pathogen, a Fusarium, and we have a bacterial pathogen, Ralstonea solanacearum, and we have a nematode pathogen, Melanogain javanica. So that is why it's called as a wilt disease complex. So whether the microbial consortium can help in overcoming this, that is why we introduced a mycorrhizal fungus, a PGPR, Pseudomonas fluorescens, and a nematode trapping fungus, Bacillomyces liliaceus. You can see the difference in seedling growth. We have planted in the field in a wilt sick soil, which gave significant difference in plant growth, nutrition, and yield. And this we have done in capsicum. This here it was in a raised nursery bed. You can see the difference between uninoculated and inoculated, and also the difference in the root growth. And this was another experiment with the microbial consortium that was done in an aromatic crop plant, patchouli. Patchouli is an aromatic crop plant, and it has got lot of importance because the perfume industry people are interested in this because they produce the perfume or the scent from this particular plant. So one of the students worked on this, and whenever she used to harvest the plant and then use it for shade drying in the lab, the entire building used to smell so good. People used to come to my lab and ask, what you are doing in your lab? It's said this, we are working with a particular plant and which is used for producing perfume. And so, and now this particular crop is, we are taking it to the field. And the reason how the perfume industry is interested in the oil content that is produced by the plant. You can see the uninoculated control, the oil content is 2.66, and with the recommended level of NPK, it is 2.92. Here, the last one with the microbial consortium with 50% of nitrogen and phosphorus fertilizer, see the oil content. This is how the industry is very much interested. There is a particular district in Karnataka, Uttara Kannada district, where this particular crop is grown extensively. Now there is a buyback arrangement with the farmers, with the industry to buy this particular crop. And here again, there's another important crop, medicinal plant, Coleus forscoli, which was introduced in the 3 southern states. It was introduced in Tamil Nadu, it was introduced in Karnataka, it was introduced in Andhra Pradesh. And with the buyback arrangement with the pharmaceutical company. So the farmers were very happy because there was a buyback arrangement. But when they planted it, what happened? There was a serious Fusarium wilt disease. Okay, to what extent? You can see the disease index was 85. Some of the farmers came with tears and then said whether something can be done to overcome this disease. And we tried with, this is a field experiment which was done in a place called Kollegal, bordering Karnataka and Tamil Nadu. And with only mycorrhizal fungus, the disease index came down to 68. When the microbial consortium with Trichoderma and mycorrhiza, it came down to 33. Again, a very common practice followed by farmers growing Coleus forscoli. And DBT gave us another project to standardize or develop a protocol for the inoculum production of fungi. And we have done this by developing the best host plant, the best substrate, nutrient solution, when to harvest the plant and all this. And this protocol is taken by 2 industries in South India, 4 industries in India, 2 in South and 2 in North, and 1 in Malaysia and 1 in Indonesia. So this has gone a long way. And people did not know how much of inoculum or where to keep the inoculum. So very simple things, but I don't know how it has not attracted the attention of scientists. And we did this, and we found that in nursery beds, keeping the inoculum or placing the inoculum 2 centimeters below the seed gives the best result. In polybags, placing the inoculum 6 centimeters below the seed gives the best result. And again, how much of inoculum has to be added? Earlier we thought 1 kg per meter square. Now we have come down to 500 milligrams. Now we are still going down, even 200, 250 or quarter kg is more than enough for 1 meter square, unless your culture must be good. That is a condition. And at this time, one more farm, Krishi Mela came. So again, we have to face the farmers. And one farmer raised and asked, we are very happy with know what has been done with mycorrhiza. And he said, I have a 10-year-old mulberry garden. Can I inoculate the 10-year-old mulberry garden? Will I get any good response? So again, I have to tell him, we have not done such old plants. So I asked him to come next year, and fortunately the sericulture department had a 10-year-old mulberry garden, and we did this experiment with them, and then inoculated the mycorrhiza in the root zone, and again with the microbial consortium, it gave as very good yield as comparable to 100% of the recommended fertilizer. So again, this is economics of mycorrhizal inoculation. This was the planning commission wanted me as a special invitee to one of the meetings, and they said, don't tell us any science. We have heard enough about mycorrhiza. What we want to know is about the economics. Tell us only about the economics. So that is how I prepared this slide, taking tomato as a crop, which we have done a lot of experiments. The yield of uninoculated tomato given 100% of phosphatic fertilizer, the yield is 9,000 kg per acre. The yield with the inoculation with the glomus leptotychum with off the recommended phosphatic fertilizer, the farmer gets 10,400 kg. That means 1,400 kg more inoculum. And the cost of tomato varies very much in Bangalore. Sometimes it comes to 2 rupees, sometimes it can go to 100 rupees. I have just taken 25 rupees for calculation. That means the farmer will get 35,000 rupees, and they have saved 50% of phosphatic fertilizer, another 1,875. So he gets 36,875 rupees more. So this probably convinced the bureaucrats in the planning commission, and that is how probably it was recommended as a area for funding by the various funding agencies. At this time, I was about to retire from the university, and I was also an expert committee member of DST. So in one of the committee meetings, one of the person asked me, what you are going to do after retirement? I said, I will work as emeritus professor in the university. That is okay for teaching, but continue your research in the soil microbiology, especially mycorrhizal fungi. Why don't you start a trust and concentrate only on research? So that is how we started this trust, Center for Natural Biological Resources. And then the first project came from DST, Seed Division. And one of the conditions of DST is that the trust must be 3 years old to grant a project. But they waived off because I was their expert committee member. So the project was immediately sanctioned with the condition. What was the condition? It was a bit annoying for me to know that. That is, they said, your students have shown such beautiful results. How many plants they have used? Maybe 100 inoculated, 100 uninoculated plants, and such beautiful differences. But will it work when you take thousands of seedlings? So this project is given just to show how it works in thousands of seedlings. The condition they put was 3,000 seedlings. One SRF, I was worried how one senior research fellow can manage 3,000 seedlings. And the Karnataka Forest Department came to my rescue. They said, don't worry, you want 30 laborers, 40 laborers, we will give. But we want to see how this technology works, because it was in the forestry seedlings. And so I was really surprised and said what my students did with 100 worked with 3000 also, and we worked with three tree species, Tectona grandis, Dalbergia sissu, and Acacia auriculaformis. You can see the difference between uninoculated and inoculated. And then we planted them. There is a degraded forest in Mandya, which is in between Bangalore and Mysore, and you can see how degraded the soil looks like. And we planted them. Again, the Karnataka Forest Department was very generous in giving us labor. We planted them, and you can see the difference of 1 year old Tectona grandis, uninoculated versus inoculated. This is in case of Acacia auriculaformis, and sorry. This is what I want to see. The one at the bottom. This is Dalbergia sissu. 52 months after planting. 52 months after planting. And this is the forest person. You can see that he looks big here because uninoculated plants were small. And here the same man becomes small because the inoculated plants were double the size of uninoculated plants. And very simple technology. I invited Dr. Vinita Sharma from DST who funded this project, and she came and I took her to the forest, and she saw this and said, why not this technology go throughout the country? Of course, she was very generous, but I am sorry that I have not taken it to all the states because of time lacuna. I don't have much time to spend in various places. And now we also from the organization, we do conduct training, come demonstration to the forest nurserymen, horticulture people, students, whomever are interested in microbial technology. So this is going a long way, and many people are getting benefited. So I started with the shortage in food in my previous lecture. So let me conclude with shortage in food at the time of independence. At the time of independence, there was not enough food for feeding the people. Thanks to Green Revolution, the high-yielding varieties of crop plants were introduced, and we were very happy because we had enough to eat. But later, after a few decades, now we are concerned about the environment because we have used heavy doses of fertilizers and pesticides for these high-yielding varieties. Now, after a few decades, now we are the concept of sustainable agriculture or organic farming has come into picture, which recommends use less chemicals and more biologicals. And all of you know that already government has made one particular state, Sikkim, as 100% organic state where they cannot use any chemicals. And I hope that it will be extended to other states in course of time. So it's here in this context, mycorrhizal fungi and other microbial inoculants play a very, very important role. Why? They are cost effective, very cheap compared to chemical fertilizers or pesticides, and they are environment friendly, and they improve soil health. Again, I think sure most of you know that the FAO is concentrating very much on improving soil health now. So soil health is given a lot of importance. When the soil is healthy, the plants growing on it are healthy, and the animals and humans which feed on these plants are also healthy. So lots of importance given to soil health. So these microbial inoculants are a boon to farmers, especially in developing countries like ours. So with this I conclude, and if there are any questions, I'll be happy to answer. Thank you very much.
Participants, please, today is the only day sir is available with us. Can you please send some questions to sir? Because he has traveled all his journey, all his career. He has shown us how many funding agencies and how many departments and how many, the numerous people he has helped, and the mycorrhizal world in India has made two wonderful fruits. Please, if you can post some questions, it will be very happy moment for us. Sir, meanwhile, I have one query.
Yeah.
This mycorrhizal, whatever forms are there, Are they same what you have applied in forestry, horticulture, and the agriculture?
Yeah. All the I'm talking only of arbuscular mycorrhizal fungi. Okay, I'm not talking of ectomycorrhizal fungi or ericoid mycorrhizal fungi or orchid mycorrhizal fungi. They are all different. They are all different. But what I have talked today, the entire lecture is on one particular group which belongs to Glomeromycota, which includes all the arbuscular mycorrhizal fungi.
Are they not host specific?
They are not host specific. As I told, no, that means we have the host preference, not specifics.
What happens in different situations like drought or water deficit when these fungi are going to stay in substratum?
Yeah, they do work under, in fact, they help in the water uptake. The same phenomenon, what I told for phosphorus, they grow much longer than the root hairs, scavenge a larger volume of soil, and then takes the water and then pump it into the host plant. As I told that one ICR project, what we have done, we have compared it with the water stress condition and the regularly watered conditions, and we introduced water stress almost 50% field capacity compared with 100% field capacity if we can take, and then even under 50% field capacity with the inoculation gave the same amount of result as 100% field capacity.
So in flood, if some flood conditions arises, whether these spores or these forms exist in the soil?
Under which conditions?
Flood or water.
Under water conditions, too much of water is not very preferable to mycorrhizal fungi. In fact, earlier literature said aquatic plants are non-mycorrhizal. Okay, and plants under water conditions are not colonized by mycorrhizal fungi. Again, we were the first to disprove that and say they do occur in aquatic plants. Okay, and so in fact I have not covered that part. That is a different subject altogether, ecology of fungi. And so now we know that under certain conditions, water, even there is water, plant present, they can colonize the plants. And that is how even in wetland rice, we could see increased growth of plants or yield with the inoculation with the specific mycorrhizal fungus.
So means the crops with I mean to say which hydroponics, if they are growing in the water nowadays, hydroponics in the organic farming, mostly the people are going for hydroponics. So this fungi will not help.
No, I won't say that. They can help. That's what I mean. Okay, they can help. That was the earlier thinking that they won't in aquatic plants are non-mycorrhizal, and that's what we have disproved, and we have shown that they do occur in aquatic, free-floating aquatic plants. What I mean by free-floating, this shoot portion is above, the root portion is below the water level, and we have also shown submerged aquatic plants, which always occur 3 feet or 3 meters even below the water level, which are submerged aquatic plants. They are also colonized by mycorrhizal fungi.
It was very interesting to listen to you, sir. Actually, it was a wonderful journey. How easily you have presented to us. It was not so easy to work on it. I can understand how much hurdles, how many difficulties you might have faced to make all these things so colorful. So, on behalf of the entire refresher course team, I would like to express my gratitude for accepting our invitation and giving such a wonderful talk and inspiring our participants. So thank you so much. In future, we will be having same kind of rapport, and we will request, and you will be again available for us. Thank you so much, sir. With your permission, I would like to end the meeting now.